So I promised you another video with regards to the human eye as far as question answering, and I'm a firm believer of attacking a subject from the other end by answering questions as well as learning the lecture material, and I find it just as important. I don't chase the material and then at the last moment tackle questions. I do it at the same time. And that helps me understand the material. These questions are based on things that I know that were asked before in past papers, and also the basic representation of the material I mentioned in my videos. And we're going to start with some relation analysis stuff, and I really urge you to pause this video and try and tackle these questions. So by all means, pause the video now. All right, so let's get started. And first of all, I'd like to share how I do this with you, not because I want you to, but because this is what I do and this is what I'm going to do now. If I have two statements, first of all, I look at each of them separately as a true or false question, and then I try and make sense of it. And what I do with true or false questions is I try to fix a sentence that doesn't make sense to me. So let's first of all examine the first statement. There are more cones than rods in the retina. Let's take a look at this. There are more cones than rods in the retina. And if I see, okay, can I fix this and make it make sense? Let's see if I write less here, less. There are less cones than rods in the retina. And I know this makes sense to me. I know this makes sense to me, so I had to fix it. So I would mark a false for this statement. Let's take a look at the next statement. Cones are responsible for color vision, and this right away should ring true. We know that color vision comes in 3 flavors, red, green, and blue. This is also the order in which they are most common. So cones are responsible for color vision, true. Very good. Let's move on. Cones require less light to be excited. Let's see. Cones require less light to be excited. Let's see if I can rearrange this to make sense. Cones require more light to be excited. This makes sense to me. Now it makes sense to me, so I had to fix it in order to make sense. So I'm going to mark a false on this one. Let's keep on going. There are more cones responsible for red color vision. And we know that we have red, green, and blue, and that this distribution is roughly around 60%, I want to say 30% and less than 10%. These are approximated numbers, and in percentage, obviously. So we have the most red. Let's see what we have here. There are more cones responsible for red color vision. That is true. True. Very good. We're going to move ahead. The concentration of cGMP decreases. cGMP decreases in the photoreceptor cell in a dark setting. Oh, that's a rather large statement. Let's see. Well, we know that in a dark setting, there's no excitation, and the ion channels are open. So dark setting, there's no excitation. Channels are open, and the molecule, the intracellular molecule that activates these channels and get them to be open are the cGMP. So there's a lot of cGMP in the dark condition. So let's see. Concentration of cGMP decreases in a dark setting. That would be incorrect. I would say concentration of cGMP, I wouldn't say increases. I would say decreases in a lit setting. So let's see if I can read it again. The concentration of a cGMP decreases in the photoreceptor cell in a lit condition, which means it's an excited condition. It decreases, and once it decreases, the channels close. So this would be true, and because I had to fix it to make it true, I would mark it false here. Very good. And we're going to move ahead. The transducin lowers the concentration of cGMP, causing depolarization. And we know that the transducin is basically the activated form, you can say, of rhodopsin. There's more at play here, but this is the level that we need to understand it in. And when rhodopsin is activated, then and when transducin is present, then cGMP decreases, and then ion channels close. Channels close, and when ion channels close, we're going from a more positive to a more negative environment, and this would be hyperpolarization. Hyperpolarization. You can actually view it as some sort of repolarization because you're gaining your polarized negative nature. So transducing lowers the concentration of CGMP, that is correct, causing the ion channels to close, and depolarization, that would be incorrect, because we know that depolarization is when positive charges go inside, And at this point, they're not going inside, so I have to fix this and write causing hyperpolarization. I'm going to read this again and see if it makes sense to me. The transducing lowers the concentration of CGMP, causing hyperpolarization. Now it makes sense to me. So obviously I'm going to mark a false here. Very good. And we're going to keep on moving ahead with the fill in the blanks section here. And I, again, strongly urge you to pause this video. So pause this video to work on these questions now. Perfect. And I know that there isn't a word bank. I like it better this way because I find it more difficult. And the more difficult it is, the more likely I'll be to be able to tackle the test questions. So let's see. The situation in which light is focused before the retina is called, and they're asking really for the two abbreviations for it. And let's see, if I have, this is the lens of my eye and this is my eye. Very good. And this is my retina. This is where I want light to focus. And it's focusing before the retina. That means here. So this is where the light rays are focusing. Well, this is short-sightedness. This is short-sightedness. And I can actually see that it's focusing before. It's focusing short of the target. It's called short-sightedness or myopia. So myopia, or also short-sightedness. Short-sightedness. Very good. Now, the resting membrane potential for receptors at the time of excitation is something. When we're talking about the resting membrane potential, we're really talking about the level of polarization. So we know that they're asking about the time of excitation. And the time of excitation, we know that positive charges can't go in. So at the time of excitation, positive charges can't go in. And when positive charges can't go in, things go from a positive environment towards a more negative environment. And this is called hyperpolarization. So we're going to write hyperpolarization, hyperpolarization, and read it again. The resting membrane potential for the receptors at the time of excitation is hyperpolarization. Agreed. Very good. Let's see. Something are more sensitive photoreceptors. So we know this could be either cones or rods, and thus could be excited in something light conditions. So what are the more sensitive photoreceptors? We know that these are the rods because they need less light to be excited. So I'll write rods. Rods are the more sensitive photoreceptors. I agree with this statement. And thus could be excited in something light conditions. So if they're more sensitive, you can say dimmer light conditions. Dimmer light conditions. Or if you don't like dimmer, you can say lower light conditions or less intense light conditions. So this is really the same thing. Very good. So something take longer to recover once excited than something. And really, I'm going to assume that because they're talking about excitation, they're talking about some sort of photoreceptor. So we know that there are photoreceptors that take longer to recover than other photoreceptors, and the photoreceptors that take longer to recover would be the rods. And I'm just going to arbitrarily put cones on the other end and see if this makes sense. Rods take longer to recover once excited than cones. I'd agree with this. Very good. And we're just moving on. During daytime conditions, we rely on something for vision, and because we're relying on something for vision, I'm assuming it's a photoreceptor, which can be exclusively found in the something. Okay, so let's see what we have here. During daytime conditions, there's lots of light. So there's lots of light, and there's lots of light. And I know that in very lit conditions, rods are saturated. They're really not giving me any information. They're constantly hyperpolarized. So cones are giving me information. And we know that at daytime we see colors. So this makes sense all the more. So lots of light. During daytime conditions, we rely on cones for vision. Very good, which can be exclusively found in the something. We already know, if we maybe we remember that in the retina we have the optical axis, and it's coming in at a point called the fovea. The fovea, and the fovea only has cones in it. Only has cones in it, and all around the fovea we can find cones and rods. But right specifically in the optical axis that focuses on the fovea, we would find only cones, which can be exclusively found in the fovea. Very good. Lowering the concentration. Lowering, what color did I not use yet? Red. Lowering the concentration of something inside the cell cause the something of ion channels and something of the cell membrane. Let's see what we have here. Well, lowering the concentration of something inside the cell causes ion channels, and we know that ion channels respond to cGMP. cGMP, ion channels respond to that. All we need to understand is the relationship. So let's see. cGMP activates the ion channel. So when cGMP is present, they are open. So lowering, so when the cGMP concentration is lowered, the ion channels close. The ion channels close. So lowering the concentration of cGMP inside the cell cause the closing of ion channels. What's left to understand is what happens when ion channels close. Positive charges can't come in, and when positive charges can't come in, my membrane potential goes from being slightly more positive to being slightly more negative, and it'll cause an hyperpolarization of the cell membrane. And one last thing I'd like to address is that in the well-discussed action potential graph, oh my God, this is horrible. There we go, something a little bit better. In the well-discussed graph, we know that this is depolarization, this is repolarization, and this is the hyperpolarization. So you may ask, if I'm going from a positive area to a more negative area, why isn't it just called repolarization? Why is it called hyperpolarization? And to be honest, the explanation makes less sense than to just remember that this is the case. So instead of getting the intuition behind this part, I just ask you to remember that when we have in the photoreceptors the closing of these ion channels, then we get hyperpolarization. We get a drop and it's hyperpolarization. And really hopefully you were able to at least address some of these questions. And if not, maybe get an intuition and understanding to what the right answers are. And I do believe this is the best way of tackling a subject. So hopefully you found this helpful, and I'll see you in the next video.